Good morning, students. Welcome to Icon Education. Today we are going to practice the exemplar questions of Chapter 8, Cell, the Unit of Life. Question number 1. A common characteristic features of plant sieve tube cells and most of the mammalian erythrocyte is. So here the key word asks the common characteristic which is seen both in the plant sieve tube cells and most of the mammalian erythrocytes. Erythrocytes are nothing but RBC. So the common feature among both of them is absence of nucleus. Absence of nucleus is the common feature which is seen in both plant sieve tube cells and most mammalian erythrocytes. Why the plant sieve tube cells are not showing the nucleus? See, when these sieve tube cells they become mature, they will lose their nucleus. The nucleus, ribosome and vacuoles are lost. Why? Because the sieve tube cells they have to give a pathway for the transport of nutrients. So a larger space should be there for the transport. There should be an unobstructed pathway. Then it can help in the flow of the nutrients. So a thin little cytoplasm will be there at the periphery. A thin little cytoplasm is there at the periphery, but the central part will be empty, which will lose their nucleus, ribosome and vacuoles to create an unobstructed pathway for the nutrient transport. Okay. And then in its neighbor, companion cells are present. The sieve tube cells, the neighboring cells are companion cells. So the nucleus of the companion cell will control the function of the plant sieve tube cells. Okay, that's why the mature sieve tube cells lack nucleus. Okay. Now, what about RBC? Why RBC they are losing the nucleus? RBC they lack nucleus to give maximum space for hemoglobin. Okay, because they should enable them to carry more oxygen. To carry more oxygen they need more space. So mature RBC they will lack nucleus to maximize the space for hemoglobin, and this losing of nucleus will make the RBC flexible by concave in shape, so that the RBC can squeeze through the tiny capillaries also. Very tiny capillaries the RBC has to pass through. So these mature RBCs they are enucleated. Enucleated means they lack nucleus, and this maturation is happening in the bone marrow. The nucleus will be lack to give them maximum space for hemoglobin, enabling them to carry more oxygen. Okay, and even the mitochondria will also be removed in the mature RBC. Then you may get a doubt, if mitochondria is removed from the mature RBC, then where from they are getting energy? So this mature RBC they get energy by anaerobic respiration. Is it clear? So this is the other explanation why the mature plant sieve tube cell and mature RBC are losing nucleus. Is it clear, students? Now let us move to the question number 2. Select which one is not true for ribosome. Not true. Incorrect statement we have to select. We know that ribosomes are the factories for protein synthesis. Okay, ribosomes are cellular sites for protein synthesis. They are responsible for assembling amino acid into functional protein chain. So we know that ribosomes are the sites for protein synthesis. If you look at the option 4, they have no role in protein synthesis. This is a wrong statement. Option D is a wrong statement. Okay, now look at the first. They are made up of two subunit. It is correct. Ribosomes is having a larger subunit and a smaller subunit. This is correct. Form polysome. This is also correct. In the prokaryote, the messenger RNA, single messenger RNA, many ribosomes get attached to the single messenger RNA. So this chain of ribosomes on a single messenger RNA is called as polysome, which is seen in the prokaryotic cell. May attach to messenger RNA? Yes, it is true, because ribosome during the process of translation, they get attached to messenger RNA. So first three statements are true. The only wrong statement is option D. They have no role in protein synthesis. Now question number 3. Which of the following is which one of these is not a eukaryote? Okay, not a eukaryote. You know, Euglena comes under Protista, Spirogyra comes under algae, and Agaricus comes under fungi. So all the three are eukaryotic except one, that is B, Anabena. Anabena is a cyanobacteria. Anabena is a cyanobacteria which is a prokaryote. So which one of these is not a eukaryote? Anabena is not a eukaryote, it is a prokaryote. Question number 4. Which of the following dyes is best suited for staining chromosome? Which of the following dyes are best suited for staining chromosome? The answer for that is, for staining chromosome, Carmine. Carmine is used for staining chromosome. Carmine is basically it is a basic red dye. Carmine is a basic red dye which is extracted from the insect colchicine, which is extracted from the insect cochineal insect. Okay. Cochineal insect. This dye, when dissolved in acetic acid, it will form acetocarmine. It will form acetocarmine. Okay. This acetocarmine is highly effective for staining chromosome because it can bind with the DNA and histone protein, and it give chromosome a pink or a deep red color, and it will leave the cytoplasm relatively very clear. So we can clearly visualize the chromosome when it is stained with carmine. Okay, student? And especially it is in the form of acetocarmine. Question number 5. Different cells have different sizes. Arrange the following cells in an ascending order of their size. Choose the correct option among the following. Ascending order means you have to go with the smallest to the largest. So we know that among these options, Mycoplasma is the smallest, because Mycoplasma measure nearly 0.3 micrometer. Mycoplasma measure nearly 0.3 micrometer in diameter. Then the second one is bacteria. Bacteria is measuring 3 to 5 micrometer in the length. 3 to 5 micrometer. Then the third one is human RBC, which measures about 7 micrometer in diameter. And the fourth one is the ostrich egg, which is the largest isolated single cell. Here one more question can be framed. Which is the largest isolated single cell? The largest isolated single cell is the ostrich egg, which measures about 15 to 20 centimeter in diameter. Okay, so 1 is option 1, which is there in two options A and B, and 2 is bacteria, that is 4, it is there in option 1. So this is the correct answer. In the ascending order, the size is Mycoplasma, then bacteria, then human RBC, then ostrich egg. Question number 6. Which of the following feature is common to prokaryotes and many eukaryotes? Now, you should, students, the question when it is given, it is something tricky given here, many. They are not asking you all. If they ask you which of the following feature is common to prokaryotes and all eukaryotes, then the answer will become different. But here they are asking which of the following feature is common to prokaryotes and many eukaryotes. So, like when you take the example of plants, fungi, okay, they are having cell wall, and some protista also. Some protistans also having cell wall. So cell wall is the common feature which is seen in prokaryote and many eukaryote. I am not telling here all eukaryote because animals will not have cell wall. So the correct answer is cell wall present. Cell wall present. Though the composition of cell wall is different in prokaryote and eukaryote, but cell wall is the common structure which is seen in both of them. Okay, if the question asks which of the following feature is common to prokaryotes and all eukaryotes, instead of many, if they give you all eukaryotes, then and in that case your answer will become chromosome present, because animal cell will not have the cell wall. Okay. Here, next option, nuclear membrane present. No, prokaryotes will not have nuclear membrane. Subcellular organelles present. No, prokaryotes will not have other cell organelles. So the correct option is cell wall present. Okay, and another question, if it is given all eukaryotes, then you can go with the first option, chromosome present. Though the chromosome composition is not same in prokaryotes and eukaryotes, but chromosome is present in both the type of cells. Then question number 7. Who proposed the fluid mosaic model of plasma membrane? This is a direct question. The fluid mosaic model of plasma membrane was proposed by Singer and Nicholson. If you look at the other persons, Camillo Golgi, he is related with the discovery of Golgi apparatus. Okay. Next, Schleiden and Schwann, they are related with cell theory. They are related with cell theory. Robert Brown discovered the nucleus in the cell. Okay. So the correct answer, who proposed the fluid mosaic model of plasma membrane, is Singer and Nicholson. Question number 8. Which of the following statement is true for secretory cells? Which of the following statements are true for secretory cells? First, you need to understand what do you mean by this secretory cell. Secretory cells are the specialized cells which they often present in the epithelial cells, and they release substances like hormones, enzyme, mucus. Okay. These secretory cells, they are the primary sites for synthesis, folding and modification of protein. Secretory cells are the primary sites for the synthesis, folding and modification of protein, because they have to be secreted in the form of digestive enzymes or hormones. So we know that rough endoplasmic reticulum is related with protein synthesis, because rough endoplasmic reticulum will have ribosomes on their surfaces and they are related with protein synthesis. So when you understand the function of secretory cell, then it will be easy for you to select the option. Secretory cells are the primary site for synthesizing, folding and modifying proteins, okay, for such as the digestive enzymes and hormones. So option B is correct. Rough endoplasmic reticulum is easily observed in the cell. Okay, so option 2 is correct. Question number 9. What is a tonoplast? This is also a direct question. Tonoplast is a membrane. It is a single membrane which is surrounding the vacuoles of the plant cell. So this is there in option C, a membrane boundary of the vacuole of plant cell. So tonoplast is the name of the membrane which is surrounding the vacuole. Question number 10. Which of the following is not true of eukaryotic cell? We have to find out the incorrect statement, not true. Which of the following is not true of eukaryotic cell? Now, if you look at the first option, cell wall is made up of peptidoglycan. Peptidoglycan is the cell wall composition which is seen in prokaryotes, not in eukaryotes. So option 1A itself is a wrong statement. Look at the second. It has ATS type of ribosome present in the cytoplasm. Correct statement. Mitochondria contains cellular DNA. Yes, mitochondria have its own DNA which is circular. Okay, circular DNA is there. Membrane bound organelles are present. Yes, eukaryotic cells are having membrane bound cell organelles. Question number 11. Which of the following statement is not true for plasma membrane? Not true. You have to find out the incorrect statement. Option A, it is present in both plant and animal cell. Yes, it is a correct answer. Plasma membrane is present in both plant and animal cell. Another question can be framed here. Which part in the cell is structurally similar in both prokaryotes and eukaryotes? Then the answer is plasma membrane. Plasma membrane is structurally similar in both prokaryotes and eukaryotes. Okay. Now look at the second statement. Lipid is present as a bilayer in it. Yes, lipids are having two layers. Okay, with the heads facing out and the tail facing in and the tail facing in. Okay, lipid bilayer. Third option, proteins are present integrated as well as loosely associated with the lipid bilayer. This is also true because two types of proteins are there, that is surface protein, okay, and the proteins embedded within the bilayer. Intrinsic and extrinsic proteins are present within the bilayer. So this is also a correct statement. Look at the fourth option, carbohydrate is never found in it. This is an incorrect statement, not true for plasma membrane, because we know that glycoproteins and glycolipids are there. Carbohydrate is found in the plasma membrane. So carbohydrate is never found is a wrong statement. Question number 12. Plastids differ from mitochondria on the basis of one of the following features. Mark the correct answer. Plastid. Plastid means they are talking about the chloroplast. Okay. So, in which structure chloroplast and mitochondria is differing? Option 1, presence of two layers of membrane. This is common because both chloroplast and mitochondria are double membrane cell organelle. Option 2, presence of ribosome. This is also true for both of them because 70S type of ribosomes are present in both mitochondria and chloroplast. Option C, presence of thylakoid. Thylakoid is present only in the chloroplast, not in mitochondria. Mitochondria will have cristae. So option C is the difference between the mitochondria and the chloroplast. Presence of DNA, this is correct because both mitochondria and chloroplast will have their own DNA. Question number 13. Which of the following is not a function of cytoskeleton in a cell? Not a function. This is also a direct question. Cytoskeleton functions are given as maintenance of cell shape and structure, support of the organelle, cell motility. All the three options are there. The first, intracellular transport. Okay, which of the following is not a function of cytoskeleton in a cell? Intracellular transport is not the function of cytoskeleton in a cell. Intra means within, transport within the cell. Then question number 14. The stain used to visualize mitochondria is. The stain used to visualize mitochondria is. It is Janus green B. Janus green B is a stain used to visualize mitochondria. It is a basic dye. It is a basic dye, and this is vitally used to stain mitochondria because it will give blue color in the presence of oxygen. Okay, this stain the mitochondria by oxidizing, appearing blue color. Mitochondria will appear blue color because of the presence of oxygen. So Janus green, this is a basic dye which is used to visualize mitochondria. So with this we have finished the practice questions for chapter 8, that is Cell, the Unit of Life. Hope you find it easy and understanding. Thank you.